Welcome to my channel. My name is Casey Charnomsky, and we're here discussing technological advancements now, the near future, the far future, and these advancements are ranging all over the board from biotech to nanotech, artificial intelligence and robotics, additive manufacturing. All these industries are going to be revolutionizing at exponential rates, and they are right now, and they're going to be affecting all of us on a day-to-day on our day-to-day lives. We're here to talk about it. We're here to talk about how they're affecting us individually and how they're going to affect the whole world once these things are out and released into the public and for people to be able to use. Welcome to the channel, and I'm looking forward to these discussions with you. This is what Cortical Labs is calling Dishbrain. This is a scanning electron microscope image showing a layer of living neurons growing on a special silicone chip at the bottom of a thumb-sized dish filled with nutrients. The chip, which is linked to a computer, can both detect electrical signals produced by the neurons and deliver electrical signals to them. What you see as the little squares in the background, the lighter squares in the grid system, those are the electrode array, and the neurons are what you see growing on top. These neurons that are growing on top of this electrode array on the silicone chip in this dish are in fact able to play and have been taught how to play a game of Pong. This was done by summer of 2022, and it was released to the public back in October of 2022. The article I'm getting this information from is dated October 14th of 2022, written by John Hamilton, titled "Brain Cells in a Lab Dish Learn to Play Pong and Offer a Window into Intelligence." Here we will see the game of Pong being played. Dishbrain playing Pong. From what I can tell, the way that they do this is they have on that array, they have the electrical signal being shown so the brain cells can be able to recognize the movement of the ball bouncing off the wall and the ability of the paddle to block the ball from getting by. It says right here it's about 800,000 cells linked to a computer. They gradually learn to sense the position of the game's electronic ball and control the paddle. In this article, it goes on to discuss how they made huge strides with silicon computing, but they're still rigid and inflexible. And Brett Kagan, the author of the study, the chief scientific officer at Cortical Labs in Melbourne, Australia, says that's something that we don't see with biology. For example, both computers and people can learn to make a cup of tea, but even basically even a computer would have any computer made today would have a hard time being able to go into a stranger's house and be able to make a cup of tea, though somebody would be able to normally walk into somebody else's house that they don't know and be able to find everything and make a cup of tea. So general information is really what he's talking about. He's talking about the difference between regular artificial intelligence and general artificial intelligence. So Cortical Labs has been trying to understand how living brain cells acquire this sort of intelligence. And Kagan says the Pong experiment was a way for the company to answer a key question about how a network brain cells how a network of brain cells learns to change its behavior. At the very bottom, he goes to describe if they hit the ball, they gave him something predictable. When they missed it, they got something that was totally unpredictable. The strategy was based on free energy principle, which states that brain cells want to be able to predict what's going on in their environment, so they would choose predictable stimulation over unpredictable stimulation. The approach worked. Cells began to learn to generate patterns of electrical activity that would move the paddle in front of the ball, and gradual rallies got longer. The brain cells never got that good at Pong, but interestingly, human brain cells seem to achieve a higher level of play than mouse brain cells. This image is of an organoid taken by Jesse Plotkin at Johns Hopkins University. It's magnified and shows a brain organoid produced in Hartung's lab. The culture was dyed to show neurons in magenta, cell nuclei in blue, and other supporting cells in red and green. The title of this article is Real AI Will Need Biology, Computers Powered by Human Brain Cells. The source is by Cortical Labs, written in neurosciencenews.com. In summary, it says here, the human brain continues to massively outperform AI technology in a range of tasks, a new study reports. Researchers outline their plans for biocomputers and organoid intelligence systems as future improvements for artificial intelligence technology. The time has come to create a new kind of computer, says researchers from Johns Hopkins University, together with Dr. Brett Kagan, chief scientist at Cortical Labs in Melbourne, who recently led development of the Dishbrains project, in which human cells in a petri dish learnt to play Pong. The article goes on to say that they're making small clusters of 50,000 brain cells, which is what you can see in the image behind me here. They're grown from stem cells and known as organoids. They get these stem cells, it says later in the article, they get them from skin samples and blood samples from patients, possibly patients. So probably donors at this point. But patients possibly in the future in order for further research and being able to work towards curing the diseases and specific diseases that those patients have. The article highlights how the human brain continues to massively outperform machines at particular tasks. For example, humans can learn to distinguish two types of objects, such as a dog and a cat, using just a few examples, while AI algorithms need many thousands. And while AI beat the world champion in Go in 2016, it was trained on data from 160,000 games, the equivalent of playing for 5 hours each day for more than 175 years. So brains are also more efficient at using energy. They're thought to be able to store the equivalent of more than 1,000,000 times the capacity of an average home computer, 2.5 petabytes, using the equivalent of just a few watts of power. US data farms, by contrast, use more than 15,000 megawatts a year, much of it generated by dozens of coal-fired power stations. So the authors of the paper outline their plan for organoid intelligence. For organoid intelligence, we would need to increase the number of neurons to about 10 million, says this senior author, Professor Thomas Hartung of Johns Hopkins University in Baltimore. Brett and his colleagues at Cortical Labs have already demonstrated that biocomputers based on human brain cells are possible. A recent paper in Neuron showed that a flat culture of brain cells, which is what I described in the last article, I don't need to go on to describe it, so what they're talking about here is they're talking about all the different things that computers are good at and all the different things that human brains are good at. Human brains are better at computers in data storage and at pattern recognition. And there's certain aspects of things that the neurons perform much better at than the computational abilities of the data processing computers that we have today. So what they're talking about is merging these technologies. They're talking about merging the ability of the neurons to be able to communicate and uh, have a group of them, an organoid of them, uh, of neurons that can get larger and larger and have more and more specific capabilities. So they're looking to data scientists and others working in computational and AI to work with them in order to be able to merge these two fields together. So we're ultimately going to be working on creating um, neural networks and artificial intelligence arrayed with uh, organoids in a neural network lattice of probably, in my opinion, a system of checks and a system of various recognition to verify AI being on the right pattern, or maybe to direct AI to be more focused on specific recognition, and for AI to go through a system of checks before neural checks before it is pushed forward. I also see a lot of this changing the debate in law about what art is, about what human writing is. If you can take a skin culture or a blood culture of a person and then take their stem cells and create neurons, and then use those neurons to then produce art, then produce writing, and then produce all the different things that a human can do. And you're talking about creating an artificial general intelligence when you're talking about merging biocomputers and artificial neural networks and learning systems. And so I believe that this is going to lead in the direction of more quickly attaining artificial general intelligence, and then as well as, if art is only classified as art if it is made by a human, then if you have human brain neurons generating that art, is that art itself created by a human? Or do you have to say, though, that human has to be able to, eat food through a mouth organ and digest it through a stomach organ? It doesn't have to have, what are the limitations to what you would label as being human? Then you get into the Prometheus ship article description. Like, at what point, when you replace and when you upgrade or when you adjust biological systems, are you no longer in the basic biological system that you started, and you're in a new biological system? Is that a fundamental change from the first adjustment, the first board removed from the ship, or does it take half the boards? Or, this is a discussion that philosophers have been talking about for millennium, and I expect that it's probably going to be something that will be hotly debated, especially here in the near future. Well, I hope that you all enjoyed these articles, and I hope that this brings up some good discussion for all of you, because I found them very interesting. Have a great rest of your day.
But wait! There's more!
This is how a scanning electron microscope works. At the very top, you have the electron gun, which fires the electron beam. Electron beam then hits the anode, which focuses it down through the magnetic lens, which then arrays the electrons in a way with a specific pattern. They shine through the backscattered electron detector and hit the image, the specimen itself, and the electrons then backscatter off of that specimen, and they have the backscatter collected from the top and from the secondary electron detector, and they're able to get a more accurate three-dimensional image than using light itself, because electrons are much more specific, smaller packets, which can have their refraction measured very, very precisely. So you can get a lot of information from using this kind of technology, you can find out you can find a lot of information about the materials, the quality of the materials, what they're made of, where there's any imperfections. You can use it for checking wires, nanoscopic wires, semiconductors inspection, microchip assembling, uh, investigating in forensics, like dusts and gunpowder and other forensic uses. It's used obviously here in biological sciences. It can also be used in soil and rock sampling. And in addition to this biological sciences, they can use it in medical science as well. So this provides a much more detailed image, higher quality image than what a normal um, microscope would provide that you can normally buy for much cheaper. The scan electron microscopes, used ones can go anywhere from a couple thousand to 40, 50,000 for a single microscope, and they're usually pretty large. Like you need a lab for them. So those are exactly how they get images with this detail and be able to provide more precise information about what they are trying to achieve.